So hi everyone, welcome to this week's sort of Exploration Club webinar. So for this week's webinar, it will mainly focus on macrophage and the microenvironment of high-grade ovarian serous carcinoma. So that's sort of the focus of today. And this sort of comprehensive analysis will be sort of focusing on using bulk RNA-seq data, particularly a specific sort of study looking at ovarian cancer patients to gain insights into the role of macrophages in tumor progression. So just to give a bit of background on how to access the platform, how to be able to run this if you were trying to do your own analysis using bulk RNA-seq data. So when you access the Biotrain ecosystem, this will be the platform that we'll focus on today, the Smart Bulk application, which can be used on internal data, or in this case, I'll be using a public case study looking at these sort of macrophages in ovarian cancer specifically. But if the user is sort of exploring it, you can directly access the Smart Bulk app. And this is where you would be able to select what species you're working on, as well as creating a new project. This could be a public data set you would be uploading or your own internal data. So this would be sort of the files that the system would require for the analysis. Okay, and this platform is within sort of the Biotrain ecosystem. If anyone in the audience is new to Biotrain and they're just trying to get an idea on where to access this analysis. Okay, so to go back to sort of the research area, so to give a bit of background on sort of ovarian cancer, if there's researchers that don't particularly focus on this area, so high-grade ovarian serous carcinoma is the most common or sort of aggressive type of ovarian cancer. It's characterized by its rapid growth and spread. And as you can see sort of in the diagram that I'm showing now, unlike low-grade ovarian sort of cancers, high-grade serous carcinomas don't typically evolve and into sort of cancers or malignant cells. So it evolves from benign tumors. The other subtypes, such as endometrioid or mucus or clear cell carcinomas, can be either high grade or low grade. And it really depends on the specific characteristics within sort of these cancer subtypes. Okay, so you can see here a bit more detail, sort of the rapid growth and how the spread in the cancer cells resemble normal ovarian cells. And this is sort of the most common or deadliest type of cancer. And this will sort of be the focus on today. So these patients with ovarian HGSC. Okay, so the other sort of diagram as well that I'll be focusing on is a bit of the background on how sort of the role of macrophages in ovarian cancer. So this diagram illustrates sort of the differences or the different phenotypes of these macrophages in the tumor microenvironment. Inflamed macrophages are characterized by their production of sort of the pro-inflammatory cytokines and their role in activating other immune cells. So as you can see, this exhausted macrophages are often associated with chronic inflammation and immunosuppression. So cold macrophages have a more anti-inflammatory phenotype, and this may contribute to tumor progression. And as you can see a bit in the diagram a bit more closely, you can see that the tumor-associated macrophages predominantly sort of display M2-like phenotype. And what does this mean? So this is normally associated with immunosuppression with the tumor sort of promotion. And this highlights, or this is highlighted by the increased production of the prostaglandins, which can suppress the immune response. So this will be sort of topics that we'll be covering today and how we can analyze the data and better understand sort of the phenotypes of these macrophages in the tumor microenvironment. Okay. So now that we sort of explored the different phenotypes of macrophages and you got a bit of background on how this plays within the tumor microenvironment, we can now sort of analyze this deeper into the specific interactions between sort of tumor-associated macrophages, other tumor cells in ovarian high-grade serous carcinoma response, and also other sort of, I guess, cell types that could be T cells or other immune sort of cells that interact within the environment. So this will be sort of the focus. So to really explore these different phenotypes or these interactions between TAMs, tumor cells, and other sort of immune cells, we'll need to sort of dive deeper into the specific molecular mechanisms involved in sort of the tumor microenvironment. So to do this, we analyzed a specific public bulk RNA-seq data set from ovarian HGSC patients. And this figure in sort of the slide illustrates the signaling network that operates in the ovarian cancer microenvironment, which is identified using the bulk RNA-seq sort of data. And we mainly wanted to sort of focus on comparing the gene expression patterns between these TAMs, tumor cells, and other sort of, I guess, T cells or other immune cells within the microenvironment. So in order to really do this and be able to sort of identify these interactions, we will perform ANOVA, which I'll show you how you're able to do that on the SmartBulk app. So we can focus on, or we can find specific genes that are differentially or co-expressed in TAMs and tumor cells. And this sort of, as a result, explores those genes more thoroughly. And this could lead us to other pathways that are likely to contribute to the complex interactions between these cells. And mainly specifically in response to sort of ovarian cancer in the specific patients that have a more aggressive form of this cancer. So to sort of illustrate some of this, I'll go into the bulk RNA-seq or the SmartBulk app. And in order to be able to perform the ANOVA analysis, we'll have to sort of upload that data set, and you'll normally be able to see the original data set that is loaded into the system. So to get an idea, this would be sort of the metadata file that you would be loading into the system. And in this case, this is the file that has all of the information relating to these ovarian cancer patients. And we can see here all the sort of metadata relating to the cell type, to the actual conditions in the patients, and all other clinical parameters that will be important when we're doing the analysis. So the next step will be sort of to perform ANOVA. And just to give a bit of detail as well, when you're performing the ANOVA analysis, since our aim is to really understand these genes that have a sort of differentially or co-expressed within the TAMs and also to other tumor cells or other T cells or immune cells. The first step will be to go to this ANOVA feature, and this is where you'll need to select the ANOVA analysis parameters. So you can select the original data that you loaded into the system. The next step will be to select what metadata column you'll be using. So in this case, since I'm trying to really find what genes are sort of differentially expressed, which ones do I see that are co-regulated or co-expressed within these different cell types. I'll select the cell type metadata. And where's this cell type metadata coming from? So these are cell types that came from the original paper that we are analyzing that I showed in the slides earlier. So this is the sort of paper that we'll be using. And the actual annotations were sort of defined using isolated through magnetic cell sorting. And from there, we standardized the annotation from the cell sorting experiment to be able to then add it as a metadata label in the SmartBulk app. So this is where that cell type annotation is coming from. From here, these are other parameters that you can keep in mind when you're uploading the data. They could be specific to the experiment that you're running. So whether there's different test options or the adjusted p-value might be a different method that you need, or what species you're also working from. So it's always important to always select what is sort of specific to your data set. Once you submit that to the database, since this does take a few minutes to load, I have prepared a sort of result here in the slide so you can see the ANOVA results. So what do we see from sort of the ANOVA looking at these different interactions between sort of the TAMs, the tumor cells, and other T cell or immune cells? So what we can see is that as shown in the blocks plots, which is sort of figures that you generate from the ANOVA, we can see that both IL-10 and IL-6 are highly expressed between both TAMs and the tumor cells. And this suggests a potential role in STAT3 signaling, which regulates the sort of interactions between these cell types. You can also note that STAT3 is sort of frequently overexpressed or constitutively activated in various cancers. So this is a sort of marker that we see across different cancers and also a sort of overly expression role within this gene. And this sort of marker has been shown to promote tumor growth as well as survival and metastasis by regulating the expression of these genes involved in cell proliferation, angiogenesis, and invasion. So this is sort of insights that we can gather from the ANOVA, especially if you're trying to really understand what markers are defining these interactions between these cells and how does this play a more deeper role within the tumor microenvironment. Also another sort of result that we can see from the ANOVA is the high expression of IL-10 in the tumor-associated macrophages, which does indicate their immunosuppressive potential. So what does this mean? While IL-6 signaling could promote inflammation, cell proliferation, and angiogenesis in both these TAMs and tumor cells, this suggests that there's a more sort of deeper complex interplay between the cytokines and the tumor microenvironment. And you can see this a bit in sort of this diagram here where we have our two cytokines and they're sort of interacting, and there might be a more complex interplay that we'll need to sort of investigate further. Okay. So if we go into the next sort of analysis, so other results that we gather. So by building upon this understanding that STAT3 induced cytokines does sort of have an effect, we can also explore another key signaling pathway involved in these TAM tumor cell interactions. So one of the other genes that was also significantly sort of found within the ANOVA result is TGF-beta 1 signaling. So as shown in the blocks plots, TGF-beta 1 is highly expressed in the TAMS group, while TGF-beta 2 is predominantly expressed in the tumor cells. And this suggests a potential role in mainly in overall in the TGF-beta signaling complex in regulating these interactions between these two cell types. So TGF-beta signaling is a complex pathway that can be both sort of tumor suppressive and have tumor sort of promoting effects. And in the context of ovarian cancer, since we are looking at this in HGSC patients, we can see that TGF-beta signaling may contribute to this tumor growth in these patients, invasion or immunosuppression. So it may have some sort of an immunosuppressive role as well. So another sort of way to think of this is that the expression of TGF-beta receptors in both TAMs and tumor cells do indicate that these both cell types are responsive to this TGF-beta signaling. And this could be sort of a potential in sort of being able to modulate both the autocrine and paracrine signaling within the tumor microenvironment. So this is another result, other sort of markers that were significantly expressed within these different tumor cells or these tumor-associated macrophages. And these are some insights we could gather from the result. Okay. So a bit to really summarize these main sort of results from the ANOVA. So what we sort of found is the key roles in these TAMs and how they play a role in these ovarian HGSC patients. So one of sort of the main aims is that we discuss that TAMs can play a complex role in ovarian cancer, and it can also influence sort of tumor progression in several different ways. One of the main ways is sort of inflammation and immune suppression. So this would be where the TAMs can regulate the tumor microenvironment and interact with other tumor cells. The second part is the actual cell-to-cell interaction role. So this would be where we see the expression of the TGF-beta 1, which suggests that these TAMs can regulate the tumor microenvironment and interact with other tumor cells. And the third, which is more relating to tumor progression, we could also see from the results from the ANOVA that the overall findings indicate that TAMs can contribute to tumor growth and also have a progression in these mechanisms. So these are sort of therapeutic strategies that we can sort of develop further. Okay, so how can we really understand sort of the role of these TAMs in ovarian cancer, and how can this provide sort of more insights into therapeutic targets relating to just the macrophage subtypes? So to further understand this in the context of ovarian HGSC, we will next dive into another sort of part of that data set from the same author, which tried to really understand the heterogeneity of these cells within the tumor microenvironment. Okay, and this data set that we'll be focusing on here is looking into these TAM subtypes. So again, it's sort of an additional study from the original author, the original paper, that really tried to identify these differences in these TAMs in these patients. Okay, so in order to really do that, we'll go into, again, the SmartBulk app to show you how we would be able to run this analysis. And this specific analysis will require sort of differential gene expression analysis. So this feature you can access within the data set that we'll be focusing on. And this is where you'll need to sort of decide what is your analysis parameters for the experiment. So what data set you'll be choosing, whether it's the original data or normalized data set. And what does this mean? So in the original paper, the author did explain that they applied TPM normalization according to sort of the sort of strategies. And this separation in terms of gene counts in subtype A and subtype B indicated that the two subtypes are not only different in expression, specifically IL-6, CD163, and PICO-CLE2. So this is sort of the method that the author performed. And a way to do this or be able to validate this and replicate this on SmartBulk, you can go to the quality control step. And this is where you're able to apply normalization using the same method that the author used in order to be able to really find these representations. And you can also color it by the actual subgroups or the subtypes. In this case, subgroup A and subgroup B for these TAM or these tumor-associated macrophages. And this is what the author performed. So you're able to really replicate all of the steps of the author, the methodology, so that you can actually run further analysis on the same type of normalization method. So this is where, again, if you want to then go into the differential expression analysis, you need to choose the data set. You need to also be aware of what statistical method would be required for this analysis. So in this case, we'll be running EDGE-R. But you can also use DSEQ2 if it's raw count data, or if it's normalized data, you can also use LEMA, which is specifically for normalized methods that are applied on the study. Okay. The second step would be in the study design, you need to make sure what your primary factor is. So in this case, since we are trying to really determine these differences in these subtypes of macrophages, my sort of group would be the subtype 2, and I would want to perform a pairwise comparison so that I'm able to really distinguish sort of the differentially expressed genes within these two profiles in these TAM cells within ovarian cancer. So this is how you're able to prepare the study design. You can perform normalization before if you want to replicate the results in the author's paper, whichever method they normally use, or you can decide to run your own analysis on raw count data as well and apply the statistical method that is specific for that. So once you submit that analysis, you're again able to see the analysis, but I do have this information prepared here as well, since it does take a few minutes to load. So in the differential gene expression analysis, you're able to see both the DGE results. So these are all the genes that are differentially expressed within my subgroup B and subgroup A of these tumor-associated macrophages. I can also sort of adjust the FDR score, so I'm able to see those markers that are significantly differentially expressed. And I can also sort of change the log fold change as well. So you can sort of be able to replicate specific results from the platform. And you can also generate a heat map result, which maps all of these differentially expressed genes across the groups. So in order to really find those particular genes that have an effect that are sort of leading to these different subtypes of these macrophages, I did prepare a slide here that kind of highlights these specific genes that have a specific role in sort of these different sort of tumor microenvironments. So in this analysis, what we're able to see is that subgroup A is mainly characterized by high expression of IL-6, CD163, and PICO-LCE2. Subgroup B, on the other hand, you see that it's characterized by low expression of these genes. So you do see that in the heat map, these very distinct clusters, and these are based on these sort of markers that are specific to these groups. So these subtypes do likely reflect very different functional states and may play a distinct role in tumor progression. So it really depends what sort of cells we see in these patients and how can these be modulated in terms of treatments that will be specific to that patient. So in order to really understand this even further, be able to sort of understand the functional states and what pathways may play an active role in shaping these cells, whether they're proliferating or they're differentiating, what you can do is perform an additional enrichment analysis from the DGE results. And what is that going to do? So that's going to give you a clear separation between subtype A and subtype B, but it'll provide you sort of a more understanding if there's any other mechanisms or functional pathways that might be driving these differences. Okay, so in order to really do that, and these will be some results I'll cover in a bit. So we'll go back into the SmartBulk app in order to show you how you would be able to perform that additional enrichment results. So in the DGE results, you'll see that we have this feature here, the new enrichment analysis. So this new enrichment analysis would let you perform additional enrichment on just these differentially expressed genes. But in this case, we're not trying to just do it on these differentially expressed genes. So what we want to do is also perform sort of an additional, I guess, ANOVA or Venn analysis. So on here, you can also select what groups you want to perform to find if there's any co-regulated genes. But if you just want to stay from these differentially expressed genes, you can just perform the enrichment test from here. So this will allow you to select the groups and sort of select what you want to perform next, or if you want to include other groups in the analysis or covariates, you can also do that as well. And what do we see when we perform that additional enrichment analysis? What we see here, if I go into the results, is that we can see that there's a clear separation between subtype A and subtype B, but it's mainly based on the immune response and the defense mechanism pathways in subtype B. So we do see that these sort of mechanisms, when we analyze the GAL and the reactome or the dose pathways, we're able to really define that this subtype B has these specific defense mechanism pathways. And these findings further sort of explain that there is a complex role in these TAMs, specifically the subtype B in these patients, that may sort of give insights into more protective or suppressive role in the immune response. So these could be really important results that can give you an idea if these can be immune modulated or if they require other types of treatment, because these are sort of more specific to defense mechanisms. And you can see that enrichment based on the enrichment test that we performed from the DGE. Okay, so to sort of go into some of the summary sort of steps that we performed in today's session. So what were we able to do from just looking at the ANOVA, looking at specific significant genes and how they're different across these interacting cell types? So we could see that the TAMs interact with other tumor cells, and this was very clear when we did the ANOVA sort of test across that data set. And we were able to define a very important role in how they shape the tumor microenvironment, involving in both anti and pro-inflammatory processes. So we were able to sort of distinguish specific markers that have these two sort of roles in the tumor microenvironment. The second step of the analysis was from there really being able to determine how these different types of tumor-associated macrophages sort of develop and if there's any differences in the functional states of these cells. So these two subtypes of TAMs were shown to have or expose contrast roles. So subtype B showed a sort of enrichment of these defense and sort of defense response pathways, which may suggest better survival or a better sort of specific targeted therapy for these specific functional roles. So these are two very important insights that we gathered today from doing sort of a validation or replication of the results from the paper and adding more insights into that analysis as well. Okay, so I guess since we're sort of at the end of sort of the session today, I will give some moments so that my colleague can send in sort of the feedback test. So you're able to give us some feedback if there's any topics you would want to see. So in this case, we covered ovarian cancer using the SmartBulk app. But you can use the poll to really just let us know what other case studies will be sort of useful for you to see how they can be run or analyzed on the platform, or if you require any specific one-to-one training using your own data. So those are support services that we do offer as well. One thing before I go into the Q&A that I do want to mention is that we are, again, running really big curation programs, both for Visium HD and also bulk RNA-seq data. So if any scientists that join the session today find this platform really useful, but they're not generating their own data and they would want to use it on public data, you can submit those requests to us, any data set that you would want us to curate so that we can deliver that for you and you can do further analysis on the platform. Especially using other new features that will be updated in the next release of the SmartBulk. So that will include WGCNA, network analysis, pseudobulk, cell type annotation analysis. So those would be sort of features that will be available soon as well. Okay, so I'll go into the Q&A section just to see if there's any questions from the team here. So one of the questions I see here is, do we also see this sort of elevated expression of STAT3 to confirm this STAT3 induction by IL-10 or IL-6? So yes, using the ANOVA analysis, we also notice that there's a significant elevation of STAT3 expression in these macrophages and in the tumor cells. So we do see this when we confirmed it using the ANOVA. And another question as well in the chat that I do see is, are these two subtypes or are these two subtypes migrate differently to the TME, or are they only differentiated in the TME? So in previous studies, I can say this has shown that the major origin of tumor-associated macrophages in ovarian cancer are derived from the resident tissue macrophages. And these TAMs are activated or skewed by different factors of the tumor microenvironment. So they do adopt a specific spectrum of phenotypes, I would say, and they do represent mixed forms that are alternatively sort of activated or sort of immune regulated, or pro-inflammatory macrophages all are also within that environment. So however, in this specific origin paper, the authors did not perform any further analysis on the origin of these two subtypes. We can use some features in SmartBulk that would allow us to run additional analysis on those subtypes, and we can perform further enrichment or perform covariate analysis if you have other metadata as well in the study. Okay. So I don't see any specific other questions here. And I think my colleague also is part of the SmartBook team will be answering some of these questions as well. If you have questions that are very private or specific to your data and you don't want to share it in the group chat, you can always reach out to support. And any of our colleagues in the team who also sort of support on SmartBook will be able to answer those more technical questions or specific case study questions to your data. Okay. So thank you everyone for joining, and I will give a few minutes just so that you're able to finish the poll that my colleagues sent in, just to get a bit more feedback on if you found this sort of session useful or if there's anything that you would want to see from the platform. And again, the platform does have many different features. If I go back to the ecosystem so you can see, we have many features here that we can cover and go through one case study, whether it's spatial data using the Lens platform, multi-integration of different spatial images as well could be another focus that you might want to see, and how that can play a role in your analysis, or BioStudio, which is more bioinformatics specific. So if there's any notebooks that you would find useful, like CellChat, being able to run deconvolution on spatial data, that would be another platform that is more for bioinformatic and AI modeling. So that if you have any specific needs that would require that module, just let us know, and we're happy to support you on that. Okay, so thank you everyone who joined the call today. I'm always happy to see those same users join every week. So we'll sort of wait for you on the next session for next week, and I'm hoping everyone can join as well. We'll be sort of circulating what that topic will be for that next session.